you find a lot of false positives. So the examples I showed you were nice examples. And what has been done a lot is, so the method is totally unsupervised. So you just take the data, you find the couplings, and then you say the biggest should be contacts. But this could be totally crazy because we know we have a lot of structures. So let's do this coevolution plus the real structure, coevolution plus real structure. In the cases where we know the answers, do some supervised learning, and this is now done typically with deep convolutional neural networks. And then you can improve actually by structural supervision the contact map prediction. There's a whole list of tools doing this, in particular for prediction of contacts inside single proteins. And probably the most known is the one by DeepMind, because DeepMind is more known than, I don't know, Jim Book-Su or David Jones. And they have done this AlphaFold, which went to the press about 2 years ago. But there's a problem. You need huge training sets, and these deep networks typically have a lack of interpretability because you have these very complicated structures and the parameters are hardly to interpret, but it's working very well. I'm a statistical physicist, I'm not a bioinformatician by training, so I would like to have something interpretable and something which works also on a small training set, because I was talking about protein-protein interaction and there is much less data. And so what we have used is recently in a paper which is on arXiv and bioRxiv, but not yet published, is that we have tried to do something which is not as powerful but fully interpretable. And the idea is basically that the contact maps are not random patterns, but they show some very specific patterns. For instance, here we have beta sheets. You have some parallel and anti-parallel beta sheets. You have, if you look to helix-helix contacts, you see some periodicity. If you have a contact here, then at about 3-4 amino acids from this, typically you have another contact because you go one turn of the helix, whereas at 2, you are on the opposite side, you typically have no contact. So there are very specific patterns. And if you look to the what is given by the DCA scores on the same proteins, on the same distinction of the data, you see it's looking almost the same. So they reflect each other. So you can implement, you can do your DCA stuff. You can use these filters which come from structures. You can combine the score and the filters and get with some very simple logistic regression. So something which is very, very interpretable. For sure not the most powerful, but very interpretable. You can get an improved contact prediction. The resolution of the picture obviously was fantastic for this to be projected here, but the black line here is just the PLM DCA for big alignments and intermediate alignments. And if we use these filters, you can get, if you use bigger and bigger filters, you can get better and better in the prediction. And here, this line is quite funny. This is one of the deep network predictors, which in this case actually worked less well. There are others which work better, but they are trained on partially overlapping data with our test set, so we cannot really plot it in here without doing something weird. So in principle, you can get more information than just sorting by size, which is good. Okay. I have to drink some water. So to do this, let me maybe go back to this slide here, or to this slide. To do this, you need actually a joint multiple sequence alignment of the proteins. So you need something which is not just one protein in each line, but which is a pair of interacting proteins in each line to actually find these couplings between proteins. But I told you that there is this intermediate problem that if you have paralogs here, and if you have paralogs here, you don't know who is interacting with whom a priori. So can we actually use the same kind of methods, similar information, to get also some insight into who is interacting with whom? So there is some intermediate step. So the examples I have shown to you, the alignments were constructed because actually the proteins were co-localized in operons, so they were always close to each other, the genes, for the interacting proteins across genomes. Okay, can we use this information? And this is something which we have done with Andrea in the first place, and I will show you some new stuff we are adding. Here, if we have this problem that we have many paralogs per species and so on, and we want to match them, so an idea is the following. So if we have the right matching, so if you put actually the interaction partners together, I hope I convinced you that there is frequently some co-evolutionary signal between the two. So you will find that there is some non-zero coupling. Now imagine the opposite case, that I make a totally random matching. You probably will have a very small, because you have no correlations between the two, or almost no correlations left between the two, you will have very small couplings. So why not using a procedure which actually maximizes the inter-protein correlation or coupling somehow computationally? It's relatively complicated. I will show you on the next slide some idea how we did this. It's a very complicated space because you are optimizing over a space where you have the species, the number of paralogs in the species factorial, which is all the possible matchings, to the number of species. So it's an enormous number. It's I don't know, the biggest number in any optimization problem I passed in my life so far. But you can reach some 80–90% of accuracy of predicting some test case, which I will show you in a second. So the idea is what we do takes into account also that the number of paralogs in the species is not necessarily the same on the two sides. So sometimes you have, for instance, here 3 and here 4. And so what we do, we always match from the smaller to the bigger one, do some injection. This is some computational facilitation, which actually probably does biologically some mess sometimes, but that's what we were able to do. And then what we do is we sort families by the number of potential matchings which are in the family, and the ones which have very few, imagine you have one protein and one protein. There's just one matching. So put these first, then do those with few and so on, and iteratively build up this by generating some initial matching, calculate models, match the next larger species and so on, calculate new models, and maybe you do something which is good, which is a progressive parallel matching. And if you want, this is a bit similar to what is done in multiple sequence alignment. So this looks a bit like progressive multiple sequence alignment, but then in many cases you can do by recorrecting part of what you have done by taking out species, recalculating them and so on. And you can do some iterative procedure which actually improves the prediction. Here I've shown for one case, which is our preferred study system, which are histidine sensor kinases and response regulators in bacteria. And here, this is a curve this is a fraction of included sequences. So our algorithm starts here and works until we have matched everything in this progressive procedure. And the best algorithm would obviously be here on this line, on this diagonal, which is the fraction of included sequences is actually the fraction of correctly matched ones. So all we do is correct. But obviously we remain below this, but we don't remain so much below it. Here we are at something like 85% still of good ones. And this 85% actually is okay. This is a protein which I showed you in the slide where I had contact prediction. So here, this is a contact prediction of the perfect solution, which we know from other means. This is a contact prediction which we get from our matching procedure. The PPVs are given here by the black and the red one. And you see that due to the errors we do in the matching, we lose a bit of accuracy, but not much, in this very specific case. But if we go to other cases, it gets a bit more messy. So now, having this in our hand, we can go back to what I told you in the beginning. and we can look to these predicted relations. So we can take these relations, and instead of having them represented only by 0, 1, presence or absence or presence in a genome, we just take the sequences and we do this matching procedure and look if there are big couplings or not. And that's what's coming out of the first 500 strongest phylogenetic couplings. So this is basically most of the tail which we introduced, which I showed you before. And the ones, actually I should say the ones which are not in the positive set. So only the ones which correspond to what is not in the positive set here, the first 500. And you see that there is some region where actually you have relatively large alignments. So you have enough data to detect coevolution. You have large couplings, so we have empirically defined some score before, which is something like 0.2, whatever this means, and you have some points which are up here. So our conclusion or our prediction would now be these are protein pairs or domain pairs where we have this large phylogenetic coupling, so co-occurrence. We have a large interprotein residue coevolution, and we have large data sets. So there we would have some confidence that maybe our predictions are good. It's a quite limited number of points up here. We have looked into them one by one, and we, as our preferred biologist for protein-protein interaction, so who knows all the stuff we don't know at all, and he has found that some of the cases were actually known because were not in the databases we used, but many other cases are plausible things, but there's no experimental proof so far that they are interacting. So it's a real prediction. But there is a lot of stuff down here and so on. And if the absence of signal does not mean the absence of interaction. It just means we are not good enough to find it, or we are not good enough to disprove interaction. We have no information. So can we do better? And so as a very last point, I want to do something which we do at the moment with Carlos, who is a visiting student in this InferNet project coming from Cuba, and with Anne-Florence Bitbol, who used to be in Paris but now moved to Lausanne. And what we use is actually the data contain not only one kind of information but several kinds of information. There's the one I discussed all the time, which is a co-evolution between residues. But there's another kind of information, and this is the information which is given by the phylogeny. If you have two proteins which interact, and in another species you have a close-by protein here, and you have a close-by protein here, there's a higher probability that these interact than any protein which is more distant.
Any residue?
Huh?
Protein or residue?
Protein. The phylogeny is at the level of proteins. So basically, the point is they evolve together, and if you go to a close-by species, the paralogs which are close to the known interaction pairs will be most likely, so they will be the orthologs of this one, they will more likely interact. There's actually a method which is built upon this, which is called mirror tree method, which is developed by Florencio Pazos in Spain, which I cited in the very, very beginning. I forgot to cite it here again, but they showed that for interacting proteins, the phylogenetic trees are particularly similar. So let's try to use this together, and so the idea is that we construct similarity networks. So now we have different species, and for instance, this green one in one species is similar to this blue one on this blue one, but it's not similar to this gray one. So these are big networks. And then we try to align them. So we try to align them in a sense that we, for instance, here this green one is there's only one green one and one green one, so we have to match them in our matching procedure. But then, for instance, look in the gray family, this gray is similar to this green one, this gray is similar to this green one, so better to match this gray to this gray than this gray to this gray.